I'm delighted to be here and to contribute to what I think is really a UK sovereign asset. I mean, the impact of the UK Biobank on advancing knowledge of human health and disease is huge, and this session is really about dementia. What I want to do in the next 8 or 9 minutes is really tell you 3 things. I want to frame the problem we're trying to tackle. I then want to tell you something about the Dementia Research Institute. And then finally, how might the Dementia Research Institute work with other national assets that Nadeem has touched upon so that together we can improve outcomes for people living with or at risk of dementia. You've got a sense of the scale of the problem. We all know that. But another way of thinking about this, today in the UK, which is not a particularly big country, there's about 1.5 million people living with a progressive, incurable neurodegenerative condition. So that's what we're trying to address. So the context of this, I think, is this area of regenerative neurology, which I regard still as an emerging medical discipline. The problem today is there are too many neurologists, like me, that essentially what we do is we chronicle demise, and we're unable to intervene in a way that is meaningful and relevant to people living with these various conditions. And this is true across all the brain disorders across the life course, which is a global problem. So now, regardless whether you're thinking about brain disorders in early life, midlife, or later life, there are some commonalities. The first is very blunt diagnostic tools. And the second is, almost without exception, the absence of meaningful disease-modifying treatments. And you've heard of that from Nadeem already. And what you'd want are treatments that will slow, stop, and if you're a real optimist, restore or regenerate that which has been lost. Now, there are some standard exceptions to this, but by and large, all the conditions that we're familiar with, we have no disease-modifying treatments. We have, for some, symptom management. Even that is pretty poor. So what we really want is we need to move to an era where we can have personalized prediction, personalized prevention, where we're thinking about predict, prevent, protect, promote. And then for those people who have got a diagnosis, give them a precise, granular diagnosis with a reasonable sense of personalized prognostic indication and what treatment options might they have. So dementias are a mixed bag. You've heard of that already. The reality is if you come to the clinic, you see me or anybody else, our precision in terms of the diagnosis is very, very poor. It is blunt. And that this graph, this pie chart is a mirror of what you've seen for Naomi. The truth is the gold standard today remains pathological diagnosis. The match between a pathological diagnosis at death and the clinical diagnosis is apart. It's not overlap. So that's the problem. And if you've got this level of disease heterogeneity, you're just not going to get anywhere in terms of diagnostics or treatments. So the question I'm often asked, sometimes in less measured language than this, is why, despite decades of research and billions of dollars, are we so far behind in neurology and brain therapeutics? The reasons are many, but fundamentally it's because the brain is the last area of human discovery, and we simply have an inadequate understanding of the working of the brain in health, physiological health, never mind disease. That is the problem. And the reasons for this are many, but essentially they could be summarized as you're dealing with arguably the most complex organ in the human body by a country mile. And the paradox is you've got the most complex organ and yet you've got the least sophisticated tools to study it. We cannot take a biopsy, we cannot put a camera and directly visualize. We're having to infer everything from imaging or pathology. That's useful to a point. We've had an over-reliance on animal models, and if there is one organ where there is major interspecies difference across the scales, it's the brain. The heart, the liver, the kidney is essentially a facsimile between a rat and a human. The brain's a different ballgame. So these are the reasons. So what we need to do, and Sidney Brenner put it very well, is we need to study man. And that is the organ, the human experimental system. And that's why the UK Biobank is such a powerful resource. There are 2 objectives really for us. We need better clinical instruments to identify people at risk, because there's a long prodrome. And then, because of this issue of disease heterogeneity, we need to be able to subtype the actual type of dementia. That's what's needed. And you need to do this in a way that is meaningful, practical, and can democratize this so it is scalable. This is a global problem. It needs to be relevant to the UK, but to the world. So the UK Dementia Research Institute is a national MRC-anchored funded institute founded in 2017. It's across 6 universities, has about 900 researchers, and our fundamental mission is through research to transform the lives of people living with or at risk of dementia. Like any serious research endeavor, this isn't done in a vacuum. It is done through collaborations, alliances, and in partnership. In many ways, we could be regarded as the science discovery engine, if you like, of a wider integrated UK dementia ecosystem. We cover all the major dementia subtypes. And it'll be obvious to you that whilst there may be different diagnoses, there are common underlying dysregulated pathways, which is the particular focus. We're rooted in fundamental discovery, but there's an increasing prominence on translation. And this brings me to the next slide. So the Dementia UK Biobank, and you've already heard from Naomi, the prediction is by 2027, there'll be about 40,000 dementia cases. But this is dementia as a label. It's a very blunt label. Even with the GP records, even with the medications for symptom management, it will not be accurate, won't even be close to it. So we need to do much better, much better. So at an estimate is half of these cases, we don't have a subtype. I would extend that. I would challenge the precision of the majority of those diagnoses. So what we need to do is we need to be able to subtype the diagnosis in a way that is cost effective. How might we do that? Well, one way is, if you could imagine, hauling back all these people who've got the label and undergoing detailed clinical characterization. Yes, of course you could do that. Paul will talk about that in other ways, and that's something we need to think about. But in the end, the problem is much bigger than this. We need to use our understanding for the biobank so that we can then scale it across the UK community and make it relevant for all people, regardless of socioeconomic status and ethnicity, which is an issue in this country, never mind globally. There are 3 areas that I just want to touch upon. We might discuss it in the panel discussion. The power today now of blood-based biomarkers to advance our understanding of disease is huge. And in the UK DRI, we've made a major strategic investment in biomarker discovery, led by Henrik Zetterberg, who's a black belt in this field globally. Today's biomarkers are comparatively crude, but they're beginning to move, and you will know that these key diseases have pathological signatures. Often it's a misaccumulation of key proteins, TDP, synuclein, tau, amyloid, increasing evidence of dysregulated immune response. All of these can now be picked up by blood through biomarkers. So imagine a Guthrie, a dry blood spot, and then people mailing that out. That's what you could do. Digital, we're all familiar with digital. There's 2 spectacular studies this year which have harvested UK Biobank data, but this is in terms of predicting people at risk, either using accelerometer data or ocular data. But those same data could well be useful for stratifying people. And then, of course, you can use digital phone-based measures, and we're familiar with this after COVID, where you could look at digital games, cognitive games, and you could imagine a multimodal integrated algorithm that brings together blood-based biomarkers, digital tests, and potentially genetics. And again, the genetics of dementia is still rooted in risk. So this is about identifying people at risk, but we're likely over the next decade also to begin to identify genes that will determine response to treatment and or resilience. So in summary, these 3 different areas, genetics, blood-based biomarkers, and digital, put together will be very powerful, scalable tools to begin to identify people at risk and or stratify patients with diagnosis. Thank you very much.